DNA polymerase can only copy DNA in the 5' to 3' direction by attaching a nucleotide at an existing 3' hydroxyl group, making it impossible to synthesize the lagging strand at the very end of a chromosome. As a result, chromosomes get shorter and shorter with each replication until reaching a critical limit, at which they exit the cell cycle and undergo senescence and cell death.
DNA polymerase copies the leading strand as helices unwinds the DNA downstream of replication fork. To copy the reverse strand, DNA polymerase has to play catch-up, creating the discontinuous set of Okazaki fragments.
At intervals of about 150 to 200 nucleotides, RNA primase adds a primer, which DNA polymerase uses to copy the lagging strands back toward the replication fork. Eventually, helices reaches the end of the chromosome and the replication fork disassembles.
The RNA primers on the lagging strand are removed by RNase, and the gaps are filled by DNA polymerase and joined by DNA ligand. But there is a problem. The leading strand has copied far out ahead of the lagging strand, with no way to initiate an Okazaki fragment to complete the lagging strand, leaving an overhang of several hundred nucleotides.
Without some way to prevent it, DNA repair mechanisms would interpret this single-stranded DNA as a double-stranded break and attempt to repair it by joining it to the end of another chromosome. The terminal regions of a chromosome are called telomeres, and they are protected by a 6-protein complex called shortening, composed of TRAF1, TRAF2, LAP1, PIN2, TPP1, and POT1.
Telomere contain multiple repeats of the sequence TTAGGG, providing a target for the vulnerable single-stranded overhang to loop back and nestle within upstream copies, forming a T-loop through stranded invasion and formation of G-quadruple plexes. The telomere sequence is initially 5 to 15 kilobases in humans, but shortened again and again after each cell division.
Eventually, the telomere region is degraded, leaving the chromosome ends vulnerable to fusion and loss of genetic material. Cell division ceases after 40 to 60 divisions, known as the Hayflick limit. However, stem cells and germ cells can continue to divide through telomerase, a specialized ribonuclear protein capable of reverse transcribing DNA from RNA.
The RNA template within telomerase hybridizes with the overhang sequence and synthesizes additional telomeric repeats, allowing the cell to continue to divide. As telomere shortening also affects cancer cells, most cancers develop a way to reactive telomerase.
For example, mutations in the promoter region of the telomerase reverse transcriptase gene or TAT may create new binding sites for transcription factors or disrupt binding sites for repressors, allowing tumors to continue dividing well beyond the normal limits. However, it also makes them vulnerable to drugs that block telomerase.
Telomerase inhibitors can act by down-regulating telomerase or stabilizing G-quadruplex to prevent telomere from being extended. Imetelstat is a first-in-class oligonucleotide telomerase inhibitor that binds with high affinity the RNA template region of telomerase, preventing priming of new telomere repeats and triggering cell cycle arrest or apoptosis in repeatedly dividing cell.
Based on the result of the IMI phase 3 clinical trial, the FDA approved the use of imetelstat in June 2024 for adults with myelodysplastic syndrome who meet specific criteria. Most other such drugs remain in the preclinical phase due in part to concern about safety and the need for long-term treatment before telomere shortening impairs tumor growth.
However,
Newer drugs such as the nucleoside analogs 6-DO2', the oxyguanosine work by inducing the formation of dysfunctional telomeres that lead to cell death through telomere uncapping.
Although progress is slow, telomerase inhibitors and other telomere targeting agents hold their great promise because they attempt to do what few other drugs can: rob cancer cells of their immortality.